Good afternoon. Good afternoon and welcome to our 2024 research briefings. Every year we take this opportunity to hear talks by newly elected members from each of the 6 NAS classes. Think of this as a sampling. The speakers represent their sections and classes, but we've asked them to focus on their own careers, telling us about their science and how they became scientists. All were elected in 2023 and formally inducted yesterday evening. I think you're going to find this session both enlightening and enjoyable. Our first speaker is Jeffery W. Kelly, Lita Annenberg Hazen Professor of Chemistry at the Scripps Research Institute. Dr. Kelly's laboratory studies chemical and biological influences on the competition between protein folding, misfolding, and aggregation. His work has fundamental importance in understanding a variety of human diseases and developing therapeutic strategies for their treatment. Amyloidosis is one example. Abnormal aggregation of protein leads to deposits called amyloid, which accumulate in vital organs. Amyloid interferes with organ function, causing disease. Leveraging insights from genetics, Dr. Kelly's group discovered the blockbuster drug tafamidis, which slows the progression of human amyloid disease. Tafamidis works by inhibiting protein aggregation, thus demonstrating the importance of this abnormal process as a driver of amyloid disease pathology. Tafamidis was the first FDA-approved drug for this purpose. And because of that, Dr. Kelly received the 2022 Breakthrough Prize in Life Sciences for his groundbreaking work. Today, he will discuss causes of neurodegenerative diseases and how drugs that modulate protein aggregation impede the development of disease. Elected to Class 1's section on chemistry, his talk is entitled Origins of Drugs That Slow Neurodegeneration. Dr. Kelly.
So good afternoon. It's a real privilege to be here. My group and I had the incredible privilege and delight to discover the first disease-modifying medicine for a human amyloid or neurodegenerative disease. That clinical trial finished in 2011. It was another 7 years before another drug was regulatory agency approved for a human amyloid disease, and that incorporated the messenger RNA degrading drugs of Ionis and Alnylam for the same proteinopathy, namely the transthyretin amyloid diseases that I'll have more to say about in a few minutes. Despite the incredible genetic evidence in the literature that protein aggregation was important in these diseases, both the scientific and medical communities became quite disenchanted with the so-called amyloid or aggregation hypothesis as the driver for these diseases, principally because there were probably 20 or so failed clinical trials for Alzheimer's and Parkinson's disease. Fortunately, in the last couple of years, there have been incredible successes towards both of those diseases. In 2022, lecanemab was the lecanemab clinical trial for Alzheimer's disease read out, showing that it slowed the progression. And just a couple weeks ago, there was a clinical trial that was initially a failure that was reevaluated for Parkinson's disease. That was an antibody that Eliezer Mosley at UCSD discovered. And my own view of the data is that it looks quite impressive, and it may turn out to actually be disease modifying for Parkinson's. We shall see. So these diseases are called amyloid diseases for a specific protein structure that I'll define in the next slide. The most common neurodegenerative disorders of this category include Alzheimer's, affecting at least 50 million patients worldwide, Parkinson's, more than 10 million, and my lab works on the third most prominent amyloid disease, the transthyretin amyloidosis, with about 5 million patients worldwide. There are two general categories of these disorders. They can either be inherited. Typically, those diseases present earlier than the sporadic forms, wherein at least the aggregation-prone protein is wild type. That's not to say there's an additional genetic modifier. The major phenotype or symptom in these patients, of course, is dementia, defined here as the impaired ability to remember and think that's serious enough to compromise the quality of daily life. These are proteinopathies. They're mainly diseases driven by protein conformational changes. For the non-experts in the audience, remember that a protein is nothing more than a string of amino acids hung together by amide bonds. And the side chains cause proteins to condense or fold into well-defined three-dimensional structures where most of the interactions are intramolecular. What happens in these diseases, as you can see from going to the top to the bottom, you have these very intricate structures. The protein unfolds partially or completely, and now the polypeptide chain in an extended conformation satisfies its thirst for hydrogen bonding and hydrophobic interactions by making intermolecular contacts with another member of the same protein. That structure on the bottom right is called an amyloid fibril. These are very well characterized now by cryo-electron microscopy and solid-state NMR. To give you a sense of scale, a normal protein is 2 to 5 nanometers in diameter. So let's think about the size of Georgetown, right? It's roughly in miles the size of Georgetown. An amyloid fibril spans from the East Coast to the West Coast. They're gigantic structures. And of course, what this field has struggled with is correlation doesn't mean causation, meaning that amyloid fibrils are in virtually every amyloid patient. PET agents are basically used to diagnose these people, although better things, in my opinion, are coming along. But of course, that doesn't mean that these structures are the main driver of morbidity and mortality, and we'll get back to that in a few seconds here. I should have made one more point on the slide that's important. So, amyloid fibrils from a structural biology perspective are relatively easy to characterize. But in these diseases, there are a lot of different protein aggregate structures that are much more amorphous and dynamic and soluble. And for me as a chemist, it made sense from the very beginning that amyloid fibrils buried deep within the extracellular matrix are less likely to have a profound effect on biology than our soluble diffusible agents. There are a lot of these, and they're becoming easier to characterize. So how is it that all these abnormal protein conformations drive the degeneration of cells like neurons and cardiomyocytes that don't easily regenerate? Well, if you look at the center of the bottom diagram, you can have a sense for the myriad structures that are abnormal that are made in these aggregation-associated diseases. These engage normal biology aberrantly, like extracellular and intracellular signaling. These structures degrade the integrity of membranes, and so you lose the integrity of cell compartmentalization. Activation of inflammation is something that's probably a very important driver in these diseases, and there's a number of second-generation drugs that are now coming along to test that hypothesis in humans. I think Larry Goldstein was the first person to show that, and who's with us today, that protein trafficking defects along axons are prominent in almost all human amyloid diseases, and so on. So I view these disorders by a mechanism that most scientists and physicians don't like, and that is neuronal death by numerous cuts, which over decades lead to the loss of so many neurons that the ability to remember and think are compromised. The last thing I should say about this slide is I've talked initially about an initially folded protein undergoing misfolding and aggregation. About a third of our proteome is intrinsically disordered, as shown in the top left. And usually as a consequence of aberrant proteolysis and or post-translational modifications, those type of proteins can also undergo aggregation and cause some very important neurodegenerative diseases. So we started studying the transthyretin amyloid diseases some 25 years ago, not because they were the third most prominent disease. We actually didn't know that at the time, but because the tissues that are destroyed by transthyretin aggregation don't actually make transthyretin. So these are cell non-autonomous degenerative disorders. Transthyretin is a transporter protein that's found in biological fluids like the blood that bathes the periphery and the CSF that bathes the brain. So that the transthyretin protein comes out of the liver into the blood, and its misfolding and aggregation, which I'll have more to say about in a minute, forming both amyloid fibrils and a lot of other protein aggregates, destroys first the autonomic and peripheral nervous systems and the organs that it bathes, principally the heart and the digestive system. And then later on, because of protein made by the choroid plexus and secreted into the cerebrospinal fluid, the aggregation of transthyretin destroys the brain parenchyma and the blood vessels that bathe the brain from outside in, leading to dementia. This is very much the order in which Parkinson's develops too. First you get peripheral pathology and then central nervous pathology. Here it's easy to explain because the transthyretin concentration in the brain is 25 times lower than it is in the periphery. And remember that aggregation is concentration dependent, so easily explained from a physical chemistry perspective. So we believe that protein aggregation was probably the initial driver in these diseases, so we spent quite a bit of effort trying to understand the mechanism, first starting with the cell biology. So it turns out that most of the transthyretin sequences leading to degeneration are secreted at wild-type-like levels. The exception are the sequences that initially present as dementias. Those proteins are so unstable that the quality control systems in the hepatocytes and in the eye recognize them as being problematic. They dislocate those proteins to the cytosol where they're degraded by the proteasome, so they're not secreted. But ironically, those sequences are secreted by the choroid plexus into the brain, explaining the tissue tropism of those diseases. Probably the most important thing we discovered is that in order for transthyretin to aggregate, the tetramer has to dissociate. And that's a very slow process. The half-life takes a couple of days. After that, everything is thermodynamically downhill. The conformational changes are fast, and the aggregation reactions are fast. So we realized that the way in which this tetramer comes apart along the C2 axis that I showed, that's actually where thyroid hormone binds in lower animals. But in humans, those two thyroid hormone binding sites are not utilized. So it was pretty easy then to think about a hypothesis where we could stabilize that weak interface by fashioning small molecules that bind to those two unoccupied binding pockets. And we argued to no avail to the pharmaceutical industry, who thought this was a crazy idea, that this was a conservative strategy because we didn't have to know which non-native structure was the main driver of pathology. We would block all of them. That was only partly the reason they were disinterested. The main reason was, as Randy Sheckman and others know, everybody at that time was absolutely convinced that amyloid fibrils were the main drivers of these diseases. And of course, this strategy would not clear amyloid that's already present. There's no reason to believe that it would. And in fact, it doesn't. So that was the big risk that we took, that blocking newly synthesized protein aggregation would stop the progression of a disease that was already underway. Now, we call these drugs kinetic stabilizers because they engage the native tetramer, and by binding, lower the free energy of the native state proportional to the dissociation constant of the small molecule. But they don't engage the dissociative transition state, the idea that the transition state has to do with tetramer dissociation. So you can tune the activation barrier of the slowest step in aggregation to make it slower. And if you do the medicinal chemistry correctly and you have a good enough oral bioavailability, you should be able to stop this. And indeed, at the dose used now, we do stop dissociation. So this was the first clinical trial for any human amyloid disease. The woman showed on the far right is named Teresa Clejo. She comes from Porto, Portugal, where there's a large family of individuals with a polyneuropathy predisposing mutation in transthyretin. So on the y-axis is this so-called neurologic impairment score, which is how diabetic polyneuropathy trials were performed up to this point. And the thinking was that these patients progress faster than individuals with diabetic polyneuropathy. So maybe unlike all the diabetic neuropathy trials that failed, this one would be successful. So this was an 18-month trial, and as you can see from the filled circles on the bottom, those individuals treated with tafamidis had a much slower progression than individuals on placebo. And like most of these trials, it's important to study these patients for years, if not decades, after the study, because not all of these drugs read out positive in the clinical trial period, as you'll see in an upcoming slide. So it turns out that after 30 months of observation, 70% of those patients who took tafamidis benefited. Either the disease progression stopped or slowed. If you simply wait 18 months before you treat them, namely those patients are on placebo, now less than 50% of the population it benefits. This is a universal truth of every single amyloid disease trial that's been done since on the wide variety of disorders. Treatment and diagnosis early is really critical. So this is the cardiomyopathy trial. It also read out positive, and as you can see, in the observation period, it was really clearly beneficial to have started on tafamidis 30 months earlier than placebo. The transthyretin protein is also expressed in the brain, where central nervous system degeneration can occur owing to the aggregation of transthyretin there. That leads to symptoms like dementia, transient loss of consciousness, and cerebrovascular bleeding. Interestingly, the brain pathology takes about a decade longer to develop than the peripheral autonomic nervous system and peripheral neuropathy, and that's because the transthyretin protein in the brain is 25 times lower than in the periphery. And remember that aggregation is a
concentration-dependent phenomenon, explaining why the pathology is slow to develop there. Now, fortunately, tafamidis gets in the brain, and so far in patients that are being treated for almost 2 decades now, we're not seeing these central nervous symptoms. So we're quite confident that they're being protected owing to the stabilization of transthyretin by tafamidis. Now let's get back to the major underpinnings of the talk, namely, where did the 9 regulatory agency approved drugs that slow neurodegeneration come from? I've told you about tafamidis. We also repurposed a Merck nonsteroidal anti-inflammatory diflunisal for those people who don't have access to tafamidis as a transthyretin kinetic stabilizer. We call all of these drugs Category 1 drugs, and importantly, they stop newly synthesized protein from aggregating, but notably, they do not clear amyloid fibrils. So the other major drugs in this category include the messenger RNA degraders. The 2 drugs that are approved already are degraders of transthyretin. There are 2 mechanisms by which this can occur. Antisense oligonucleotides use RNase to degrade messenger RNA of transthyretin, and there are RNAi-based strategies that have been developed by Alnylam. The antisense oligonucleotide strategies have been developed by the Ionis Corporation. These drugs work against both transthyretin amyloidosis, and importantly, they've recently shown success in a hereditary form of ALS, wherein the superoxide dismutase protein aggregates. So let me tell you in the next slide how this category of drugs works. So the notion is that when aggregation-prone proteins come off the ribosome, they can misfold and aggregate into a number of structures, including amyloid fibrils, for which these diseases are named. And the idea is once you stabilize the newly synthesized protein, or you lower its concentration below the so-called critical concentration that can support aggregation, what happens now is that you allow degradation to catch up. And degradation can remove all of these non-native misassembled structures that drive neurodegeneration. Interestingly, Category 1 drugs do not remove the amyloid fibrils that have been long thought to drive these diseases. Apparently, they don't do so because these Category 1 drugs are able to slow or stop neurodegenerative disease progression without clearing the amyloid fibrils. Now, of course, that's not to say that amyloid fibrils are good. They are not. They stiffen tissue, but they're likely not the main driver of morbidity and mortality. The Category 2 drugs, in contrast, clear amyloid fibrils. There are a lot of these under development. The only drug that's approved so far for use in humans and is used regularly by physicians now is lecanemab. This is a monoclonal antibody that targets the amyloid fibrils in Alzheimer's disease, and it recruits microglial cells to the amyloid, and microglial cells are fantastic at engulfing and degrading amyloid fibrils to mediate their disappearance. And in the bottom panel in this slide, this is an amyloid PET agent, basically a small molecule that binds to amyloid. And you can see that over the course of 18 months, this drug is very good at clearing the amyloid in Alzheimer's patients. And in the top panel, what we're looking at here is the decline in cognitive function in Alzheimer's patients. And you can see that those individuals on lecanemab do better than those individuals on placebo. Now, these curves will undoubtedly flatten out as we get to earlier and earlier stage patients. In fact, That's already been demonstrated in the reanalysis of the data. So I'm very optimistic that this drug will have a profound influence on ameliorating Alzheimer's disease going forward. Now, how do drugs that stop the synthesis of newly synthesized protein and clear amyloid both slow neurodegeneration? We hypothesize that the category 2 drugs block secondary nucleation. So to put this in perspective, when we think about Alzheimer's disease, we think about the aggregation of A beta and tau, and transthyretin amyloidosis with the aggregation of transthyretin. But the truth be told that in neurodegenerative disease patients, they often have aggregation of multiple proteins. And we hypothesize that the surface of amyloid fibrils recruits misfolding prone proteins and converts them into aggregates by a process of secondary nucleation. And the idea then is that if you remove the amyloid fibrils vis-a-vis the category 2 drugs, now again, you start to clear all these other misfolding prone proteins. And we hypothesize that those are the main drivers of neurodegeneration, not the amyloid fibrils that are buried in the extracellular matrix and really not available to mediate much pathology or biology. So in summary, what I hope I have told you today or provided examples of is that protein aggregation is really the main driver of morbidity and mortality in amyloid diseases. The genetic data have been in the literature for almost 30 years supporting this hypothesis. But of course, skepticism started to arise about 15 years ago because of all of the clinical trial failures. Fortunately, we've had a lot of recent clinical trial successes, and I think it's now clear that the pharmacological clinical trial data strongly supports the notion that the aggregation of newly synthesized proteins really does drive these diseases. Now, that's not to say that there couldn't be other secondary drivers like neuroinflammation. That's being investigated very vigorously at the moment. I wouldn't have much to say today without all my coworkers who really made all this possible, and I thank you very much for your kind attention.